Thanks, everyone. I just see there like very many people. I see there Zhen from our group. Also, I see Shangyan from our group. Shangyan, thank you for switching on your video. So if you can switch on your video, that would be really great, so that we will really feel the kind of your presence here. Okay, that would be really especially when you talk, when you comment something. Otherwise, you can keep your microphone muted, so that would be also helpful to reduce overall background sound. What else I wanted to say, that after this session, that will be the first one. The next one will be presented by our Polish colleagues, so you are welcome to prepare some paper, and then when you prepare the presentation and video, you can send it to us. We will send to you the link how you can upload it, so that we can upload it also later to YouTube. And yeah, so that will be then the next presentation will be in 2 weeks. So, and then 1 week prior to that, we will have our presentation to be uploaded. So here in this room, we have Shi-Yu, Nastya, as well as our colleague Anna, and also, I'm very sorry, Wenbo, exactly, yeah, I'm very sorry. And we also have Ti-Fei. I see Vitek there. I see Olga. I see Rizwan and Imran and Jakub and Muzelian. Sorry if I spell it wrong. So welcome, everyone. So you're welcome to interrupt Nastya anytime. So if you have any questions, because this session is exactly for that. Harry, hey, great to have you. So that's Harry. Also, he's joining. No, it's no problem. No problem. It's, you join us so that we can. I think we can see you by the video, so it's perfect. Okay, Nastya, so the stage is yours. Please, go ahead.
Okay. Hello, everyone. My name is Nastya, and there are so many people here, and thank you all for your questions. I really enjoyed answering them, and I hope we will have a nice discussion today. So let's start with the first question. It's not like a question, basically. I think it's just a comment. A minor point, the second significance indicator in figure 1E refers to RS-PMS versus RS-UMS. I assume, right? The placement looks weird. Yeah, you're right. I don't know why this significance indicator just shifted to the right, but it refers to RS-PMS versus RS-UMS. Yeah. So, any questions so far? Okay. We can continue. The second question, you mentioned the increased anxiety levels in scramble versus shRNA groups at time points later after restraint stress, which is not supported by figure 4EF. There were just tendencies towards decrease in shRNA group versus scramble rats. So I think the point here is that the knockdown of OxyTR had no effects on what happened. Okay, sorry. The point here is that the knockdown of OxyTR had no effects on anxiety and depression levels in either PMS or standard rats without restraint stress. And but we can see, like as you mentioned in the question, the tendency that the level of anxiety increased after restraint stress. So yes.
Yeah, my point was that it was not actually increased. There was only a tendency, right?
Yeah, because it's not that significant.
It's not that significant. You can say it's not that significant. Be careful about that.
Okay.
It is not significant.
It's not significant.
Only a tendency.
It's not significant.
Because you said it is increased. We can say it is increased only if there is a statistically proven increase. Okay, so be careful with the formulation. Okay.
Yes. This question is about the corticosterone. Generally in stress-related experiments, do they take into account the daily corticosterone level and the time of the experiment? And if there are any information, if the corticosterone level is high, the animals can tolerate the stress, and if the animals are exposed to even mild stress when their corticosterone level is low, are the more vulnerable things? Yes, normally they should take into account the daily corticosterone level, but in this study, corticosterone concentrations were measured only at postnatal day 56 before restraint stress, then during the first day of restraint stress, and on the last day of the restraint stress. So they didn't measure the.
Wait. I think you misunderstood the question. The question, I think, was how the corticosterone level changes during the day. I don't think they mean, the question here was about how it changes from day to day, but
during the one day,
during one day. And I think they have the data about that, right? Don't they show? I think I also had a question about this very difficult, like quite complex representation about the corticosterone levels, because I couldn't decipher which one belongs to which. You see, it's in one eye. And there are this green, what is this? Gray.
I cannot open it. Let me check.
Oh, okay. And anyway, so my point is that you can.
I can try to open it on my phone to see.
But we will come to that. So because I also asked about this question, so that. So you say they checked it only on one point, right? On one time point.
Yeah, 3 times. I mean, but I guess they didn't mention that they measure the corticosterone level during the day.
I think that was the question here.
Yeah, but they didn't do this. I mean, they didn't mention it in the paper.
Yeah.
So I think
that was the question, because it makes sense, right? So
yeah,
the corticosterone
of course.
They increase just before the active phase.
Yeah, I think they should have done this, but I guess they didn't do this. So it's kind of a drawback of this paper.
And the question is again, at what time did they do this? I think Vitek would like to comment something.
Oh, yeah, okay.
You just unmute yourself and just speak. You don't need to ask everyone. You are welcome to interfere anytime. Just unmute yourself and start speaking, okay? If you have anything to say.
Okay. So another question related to the measurement of the corticosterone level during the day and night is whether these experiments were done in the resting phase or the active phase, right? It is also, I think, not written in the paper.
Yeah.
And this strongly influenced the level of corticosterone. And in general, the design of the experiment.
Yes, I agree.
Totally agree.
So
yeah.
maybe we cannot rely on this data at this point. Yeah.
No, we can rely, but it is kind of.
But we should take into account that they.
Yeah, so it could be that the outcome could be different.
Yeah. Okay, the next question. Figure 3I and 3P indicates that without RS, neither the injection of Oxtra and into the PMS group, nor the injection of Oxytocin into the UMS group significantly altered the behavioral phenotype. It seems that in the absence of RS, oxytocin is not a direct factor influencing anxious, depressed behavior in mice. So why did the UMS group show a more pronounced anxious, depressed behavioral phenotype before RS on figure 1? The article suggests that the UMS group has synaptic dysfunction in the mPFC, but what factors might contribute to this? So it's a very good question, but I think that the point was to check if the early life stress can somehow influence the ability to cope with stress and the anxiety level during the adulthood. So as we can see on this graph, on this figure 1 down below, they started these separation processes during the postnatal days 4 till 21. But then they, as I assume they gave some rats some time to grow up, and become adults, and then they started their behavioral tests. So when they did these like separation processes, of course, rats were exposed to high level of stress, which we can see on the figure 1, because the UMS rats showed the increased level of anxiety compared to the PMS stress,
and also is about this depression like behavior, about this sucrose test.
But so it's also in anxious and depressed both, not only about depression.
Like the first part of the question is the first part, like this first question as well as the second question. So this all is related to this sucrose test, which means like we need to refer to this 3P, right? And also to the 1B, correct?
1B.
I guess. Because they're asking why there is.
I think Wembo asked this question. Yeah. I think it wasn't about depression only. Yeah. It was both about depression and anxiety level. So, I mean, my point is that when the rats were exposed to stress during the early life days, of course, they showed the increased level of anxiety and depression both. But after this, I mean, maybe somehow they just.
No, but then it would be wrong, because they do show the influence.
On figure 1, you mean?
No, on figure 3, they do show that it is both the Oxtra as well as the.
No, figure 3. They are adults here on figure 3.
I know.
And so we cannot see the difference. Before restraint stress, they don't show the increased anxiety level or depression level. That's the question. So why initially they showed this increased anxiety level, and after some time.
To which one do you refer now? Like, to which panel do you refer now?
I mean, the first
we cannot see that. Like, nobody can see.
Oh, sorry. Okay. This one. Yeah. This one. They're adults here, so we can see that before the restraint stress, there is no difference in the anxiety level here, but this difference can be observed here on the figure 1 during these maternal separation processes. So that's
there is no difference. Where do you see the difference?
Between UMS and PMS? Where is my here? I mean, we can see the difference.
Which difference do you refer to now? PMS and PMS before and after, without stress and with stress, there is no difference. Where do you see the difference?
After the restraint stress on figure E and figure.
In figure 1E, the purple, the magenta against the magenta is no difference. And the same they show here in 3J, where you compare the red against the red in 3J. There is also no difference. So there is no difference.
Okay, so
it is in line. It's all in line. I think the only thing which they don't have the difference is about this depression-like behavior. And for me, the authors just dissected that there is a depression-like part of this behavior, and this part of the behavior which is related to this stress, coping with the stress. And they say that this oxytocin pathway from the PVN is overall, for the paper, they prove that this pathway is important only for that part, for the stress, coping with the stress, but not with the depression-like behavior, because there is no change in this depression-like behavior if you manipulate this either oxytocin or oxytocin receptor or this whole pathway. That's my understanding. Maybe I'm wrong, but that's how I
understood the question. Like, there is no, as you said, there is no difference on figure 1. But why is there a difference on figure 3? After the restraint stress.
Which panel do you mean?
The red one.
I don't see.
The red one. This one. So here we can see the difference.
Of course.
After the restraint stress.
Yeah. So they show that it is dependent on the
oxytocin receptor. Yeah.
That is what they show. That's the mechanistic proof.
So, Wenbo, can you explain a bit what was the question?
Without restraint you can see.
Okay.
No, you can increase the volume. It's fine.
So which one? Increase the volume of. Okay.
It's fine.
But that's a volume of. I think it's a volume of output, not input.
So in figure 1D, you can see UMS, there is a significant difference between UMS and control STD. So
in figure 1D.
Yeah, duration inside the hormone.
What to compare? What against
UMS.
what? UMS against
STD.
Against STD in control.
Yeah.
Yes, there is. Yes. Okay.
So I wonder why. Because oxytocin is no difference.
Wait. Well, what are you asking now? So you're referring to in 1D we have this green against gray, which is significant. Yeah. So they, like, if you don't unconditionally separate, then they explore less. Okay. They explore the central zone less. So what is your question?
So which factors contribute to this?
You are asking referring to which panel? You are asking referring to figure 3 or what? What is the particular? That is exactly the point. What they are trying to figure out. What is the overall? Which factors contribute to that? And they prove here that it is oxytocin, an oxytocin receptor, and it's signaling from the PVN to the prefrontal cortex. This is.
The question, sorry.
Yeah. So did Anastasiia address your question, or you are still having something unclear about that one?
I don't.
Yeah. Sure. Go ahead, Harry.
I think it's okay. I think
Have you finished your question?
Okay. Maybe let's make Wang both finish. Yeah. Yeah.
Because I see in figure 3D, 3N.
the right one.
3N. 3N. Yes. Yes.
And also OP. I just think the oxytocin level didn't significantly decrease.
Oh, it did. Just they didn't show this. I think they don't show the. You mean the green against green? You think this one?
No. Green against a black.
No. Green against black is something else. You're mixing up. So here, if you refer to 1D, the gray against green is basically showing the how's it? I'm not getting confused. So UMS before and after stress. So this is green against green, right? So the UMS generally has a low level. So that is already the low level. What you see here in this N. So they don't have other groups here. And after the restraint stress, if you don't treat it with oxytocin, it will be decreased. But if you treat it with oxytocin, it will be normal. So the oxytocin can rescue that. So they show that it is dependent on the oxytocin, and it is because of the lack of oxytocin that, or oxytocin projections, that we have this decreased coping with the stress. That's at least how I understand that.
Yeah, it's correct. I mean, I also understand that.
Okay. Go ahead, Harry.
Should I have?
No, you just talk.
Okay.
Just talk.
I have, because I'm not a human. I have not learned about human for more than 8 years. But as I still remember, I mean, like, because I learned a lot about plant. Yeah. So I have one question. Because that inside our brain, right? Actually, we have the cerebrum, and medulla oblongata, the middle part. So why they choose to test this chemical oxytocin? I mean, like, for the separated and unseparated from the maternal. And my second question, when I read the journal last night, right? So about the method they use, I mean, like, because sometimes they check for the chemical. They also did the chemicals screening test to see the resilience. When they leave their mother, they become more, how can I say, more resilient. So how does it relate to those chemicals? That's my questions. Yeah, yeah, but because that there is, I believe there is, besides oxytocin, there are still others which plays important role.
Yeah, that's true, but they just decided to focus on the oxytocin in this study because there is evidence that like in PVN, the brain region, oxytocin pathway is very, gets activated in terms of stress response. So that's why they wanted to clarify if it is so. And so they checked the particular oxytocin pathway, and then they also tried to show that this oxytocin in PVN, like also, how to say, there is the pathway from the PVN to the mPFC. So there is a whole oxytocin pathway that is involved in the stress response in humans. Yeah, so that's why.
Just now you mentioned about the cortisol.
Corticosterone. Yeah.
So can I assumption if, let's say, from this figure, right, when the oxytocin is high, means the cortisol is high, means when they leave their parents, their mother, they become more tough, become more resilient. Is that the conclusion
But they didn't.
from this paper?
No, they didn't check the cortisol. Only the cortisol.
No, but if let's say because this one is only oxytocin, because as I know, cortisol also relate to our stress response.
Yes, but they just.
They just check with the oxytocin, but
yeah.
So my overall conclusion is that the stress level generally is lower. So that's what is measured. Also, you see this figure 1L, they see lower levels of corticosterone. In rats, they have corticosterone, not cortisol, right? Not cortisol. Cortisol is in humans. It's very similar, but it's the same function, right? So what they say is that if they are kind of preconditioned like that, then they have also lower stress. So that is overall conclusion. But corticosterone is not the main line of their research here. It's just the additional parameter just to show that there is decreased stress. So that's the point. Okay, if we are through with this question, maybe we can continue with the next one.
Yeah.
Unless like anyone would like to comment anything else.
We still have so many questions.
Yeah, I know.
Okay. In addition, after restraint stress, there were no significant differences in oxytocin protein expression and behavioral phenotypes in the UMS group compared to the control group, figure 3F and figure L. It seems that the stress tolerance of UMS mice was not affected. How can this phenomenon be explained? So, if I understand the question in the correct way, so we can see the results of RT-PCR, which show that the oxytocin expression in mPFC in PMS is higher than in standard rat and UMS rats, both before and after restraint stress. And also on the graph F, we can see the results of this enzyme-linked immunosorbent assay, which also demonstrate that oxytocin mPFC in PMS and standard rats is higher than in UMS rats before restraint stress, and after it, oxytocin level in PMS is higher than both in standard rats and UMS rats. And also in the supplementary data, they also showed this graphs G and H. So we can see that the expression of both oxyTR mRNA and oxyTR protein in PMS rats is significantly higher than that in standard rat and UMS rats. So I think I just didn't mention, in my presentation, I didn't mention the oxytocin protein expression. because they only mentioned this in supplementary data. So I didn't maybe get the question in the correct way. So correct me if I...
Whose question?
My question.
Okay, I'm sorry. Maybe I just misunderstood the question.
Maybe next time just include like who
Okay, but you just asked about the oxytocin protein expression, but I didn't mention about this. So I mean, they have the data about this, but in supplementary, not in the main figures. That's the point, yeah.
Okay. But
So-
is it addressed? Well,
I don't think there is a significant difference between UMS and STD about the oxytocin expression.
On graph E or which one?
no, there is no. Yes.
Yeah, all of them, I think, didn't show the significance.
Wait, but for the
But-
PMS, it is increased, and that is
Yeah.
exactly the point.
Yeah.
Yeah, I don't care. I just want to know why UMS no significance, but
That is how it is. That's what they show. I mean, that's and oxytocin level in PFC. Actually, there is decrease in UMS in the standard before the restraint stress. There is decrease of the oxytocin in the figure F. You see, yeah, the gray against the green. There is a decrease.
but I just want to know after restraint.
Okay, the point is very clear. So the PMS kind of prepares you. So it increases the expression of the oxytocin, and this is very clear. There is no significant decline on other points. Yes, but I think this still goes in line with the message. Yeah.
The hypothesis is still...
Why it is like that? So your point is it should be done, right? Or
Yeah,
I think
Okay, I understand, yeah.
I think nature or whatever doesn't owe us anything. So this is how it is. So this is what we see. Maybe it should not. So maybe it is just like remains on the same level, and without like much stress, it is fine. We are totally fine with that. But if there is some stress, we are not prepared, and then these low levels of oxytocin cannot rescue us. But if we are kind of preconditioned, there is increased oxytocin, so we are prepared, so we can cope with the stress better, probably. Yeah. So we can speculate, but that's just a speculation, right? Yeah. Okay.
Any other questions? Or...
Regarding this point.
Yeah. I guess...
Okay.
No, okay. The results of C4's expression seem interesting and worth mentioning. Yeah, I agree. I checked the supplementary figure 2 with the C4's expression data. So yeah, they mentioned in the paper that they identified 17 brain regions that consistently displayed C4's labeling. So in the claustrum, RSD, and this 2 means tubular infundibular nucleus or something else, because the abbreviation is... Okay, it's correct, okay. And medial prefrontal cortex.
Nastya, question. Did they stimulate C4 somehow? Or it was kind of, how did they do this? Like, did they do this just, like what was the stimulus? Because in order to induce C4, sometimes you can activate, you can use some kind of challenge. So did they do that, or how did they do this?
I think they didn't mention about this much in the paper.
Okay, so if it is mentioned, it should be mentioned in the materials and methods. But okay, so I think that is also a point which deserves.
But I don't know why they don't put these figures, like main figures, in the
Because
paper.
it is not directly contributing to the line of the story, so that is fine. But the key here for
But this is like a typical attitude of many groups, that they use the C4's expression as a marker for the neuronal activity, which is apparently not the case, right? because it is rather the marker of a novelty. And the marker of a strong rearrangement of neurons, and a good example to see the difference between the activity and the novelty or plasticity, let's say, is to compare the expression of the phosphocrep with C-phos. Usually you see a lot of phosphocrep presence in the many neurons, and this is rather the marker of activity, simple activity, and compared to C-phos, which is not that abundant, and it is seen only in neurons which undergo very deep plastic changes. So. And moreover, you cannot just check the activity of C-phos like that in any time of the day or something, because it changes strongly.
That was exactly my point, yeah. So that whether they have somehow changed or challenged it somehow, then checked it after, for example, half an hour or something like that. That's one thing. Or they just check at some point, and
yeah, maybe they mentioned, but I don't remember.
At some point, and at which point, yeah. So really, thank you very much. Really great, great comment here. And for us, of course, for the story, like the main, the most important
mPFC,
part is the lowest one.
Yeah.
About the medial prefrontal cortex. Okay, cool. Nice comment, Vidik. Thank you.
Thank you. Do you know which experimental design is more often used in papers, predictable or unpredictable maternal separation? And do you have knowledge about changes in the corticosteroid system, not only CORT but also GR, depending on stress type? Actually, I think the novelty of this paper is basically that they divide this maternal separation into predictable and unpredictable ones, because usually many researchers just use unpredictable maternal separation paradigm to expose stress to the rats, like rodents. And here they showed the difference between these two paradigms. And we have knowledge about the corticosteroid system. I think I just, there are some papers that I found to show you. So actually there are many studies regarding the corticosteroid system and both glucocorticoid receptors and neural corticoid receptors as well. So yeah. And are there any changes in the corticosteroid system depending on stress type? I'm afraid I cannot answer this question because I didn't find any papers regarding this particular question. So I think there should be some differences for sure, but I don't have any data to.
And I have also one general comment, if I may. Maybe it would be good if you present on your YouTube presentation the table S1, which shows the differences between this predictable and unpredictable stress. It's really interesting, because I thought that it will be, I'm not the expert in this stress application, but at least to mice, maybe to humans, but I have more experience. Because the difference between these two paradigms is not really very huge, considering the differences in the synaptic plasticity, so the level of dendritic spines that they present, which is, in my opinion, huge, and these mice with unpredictable stress, considering these strong changes in the dendritic spines level, they should be really strongly affected. And probably you do not have this table as one, which shows the differences, but we could maybe discuss this, and if you can show it, then then you would see that the differences between these two like paradigms is not very strong.
Okay.
Stop sharing, please.
Yeah, I can.
Okay, so it was.
Okay, thank you.
Okay, I'm sharing now. Okay.
Yes, so this is this unpredictable one. And in case of predictable one, they can also the separation applied for 4 hours every day at the same time. So right?
Vidik, we are losing you a little bit. Your sound is breaking. Maybe.
As you said, this is common, the separation paradigm, right?
Vidik, your voice was breaking for around half a minute, so we might have missed some of your points. If you can repeat it again.
So in general, in my opinion, I'm not the expert, but in my opinion, the difference between the predictable and unpredictable paradigm is not that huge, considering the changes that they present. So these changes are really beautiful, very strong, and the difference in the stressors applied to these mice is not that different. I mean, that's the comment, it's not the question, but
Okay.
general comment is that maybe during the presentation that you uploaded last week would be good to start with that, with this comparing these two paradigm and discuss a bit on that.
Yeah, okay, thank you. Thank you for coming.
So, Vidik, I didn't actually check this table as one before myself. And what would you, can you draw some attention to, like, what are the key points here? Because I really didn't study it well. I didn't look into that. So so your point is that it looks like it is not really so different, right? The UMS and PMS, right? So that is the point.
But the idea, like, of the paper was that.
Exactly.
Okay, yeah, you can continue.
Okay, so my point is that for me, it is also to some extent predictable because they get this separation almost every day, except 4 days here. So it's not really that they can predict that something will go on, something wrong will go on. That's my point. The difference between the predictable and unpredictable,
which makes.
I cannot agree with you, Vitek.
Yeah, Jakub, please continue. Jakub, please continue. Yeah. Jakub, we can hear you. Yeah.
Maybe yes, maybe now it will be better. I don't agree with Vitek. Right, because as you can see, sometimes there is 3 or 2 separation of the animal by 1 day, and the biggest peak of corticosterone is in the act of separation. That's, sometimes these animals could have 2 or 3 peaks per day, or in the egg. They had the end. So in my opinion, this paradigm changes a lot in behavior, and it is pretty unpredictable and harmful for animals, in my opinion.
Okay, thank you.
I think the idea was just to separate them every day, but for the unpredictable stress, they separated them every day in the same time. But for the predictable stress, they separated them every day, but like in different time. So that was the point. They only changed the time. when they separated them.
So unpredictable is same.
Predictable is same time, unpredictable is different time. Yeah.
So the different time, do they use the random?
Yeah.
Randomly?
Yeah.
So
Like very very randomly.
So I have one question. Can you turn to previous question?
You mean slide?
Yeah.
Okay. Let me.
You can share the screen now.
Yeah.
Can I help you?
Yeah. I forgot how to caps lock or how to. Oh, okay. 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 You want which slide?
Yeah.
Sorry.
The down, the up. Yeah, this one. Because in plants, right? This one is it from confocal microscope? The result, is it also from confocal microscope?
Doesn't look like it's confocal, but in this case, it doesn't matter much.
Oh, okay. Because I'm just questioning that if, let's say, in plants, right, we want to see the protein expressions, and we use the confocal and then fuse like with YFP, yellow fluorescent protein, or GFP as a reporter gene. So like this one, the color one, I mean, like what makes it like the color? Sorry, because that I don't study about the brain, but I'm just interested.
That is what Witold was talking about. This is the sign of novelty, the way how the cell gets, it is changing its expression level. So C4 is the part of the very strong and very versatile transcription factor, which is influencing many different downstream genes.
Yeah.
So they don't need to colocalize it something. They are not interested in specific populations. They are only looking at this at in general, in the brain in general. So that is the point.
What I mean, my question is how to do this experiment. I mean, like how to do this experiment. Because
I was asking Nastya whether they did some kind of challenge before measuring that, and she couldn't answer because we couldn't see. So whether they did it at some specific point or they challenged somehow, that is a good point. So we don't know whether it was related to some time of the day or to some specific challenge, or they just bluntly just screened overall C4 expression. So that is also my question. So we don't know. Yeah.
So like this figure, right, it's not from confocal. So from which software?
It doesn't matter. You can do it in a normal fluorescent microscope, not necessarily from the confocal. Maybe it's confocal, maybe it's not confocal. It doesn't matter. You don't need to colocalize it with anything in this regard.
I'm just curious because in plants normally we study the protein expressions from the localization.
It all depends on what is your question, your research question. Yeah, so in this regard, it is totally fine to do it also without confocal.
Mm-hmm. Okay.
Okay.
I can continue. You mentioned the increased frequency of NLT in UMS and PMS, which I cannot conclude from figure 1F. So yeah, figure F depicts that no difference was observed in novel location recognition, and there is no significant difference between standard red and PMS and UMS reds. I guess I just mixed it up during my presentation. Yeah, so you're correct.
There is a question from Musi again, or maybe it's a command.
I think it's yours. No?
No. I'm saying in the chat.
I cannot see the chat because
I can read it aloud. In article it says to identify target brain subregions through which mild early life stress mediates its anti-anxiety effects, we detected C4s. Okay. Followed by open field test. How can they detect C4s following the open field test? This I don't understand. You first do open field, then you harvest the brains, and then to detect the C4s, not vice versa. That cannot be like that. That's probably a mistake. C4s labeled neurons were quantified every 120 micrometers yes, throughout the rostral extent of the brain based on anatomic registration. Thank you, Ms. Yuan, that's really helpful. According to an existing brain atlas. In total, we identified whatever they were. Okay, so it doesn't look like they did some kind of challenge, right? So except
They didn't describe anything special.
and they mixed up like that they detected C4s
Before,
by open field, or they wanted to say following.
Following, okay, maybe.
Those are the authors which are probably not English, like not
Native
native speakers. Maybe they have mixed up. Yeah, so probably that's a typo. Following by open field test. That's what they wanted to say. Okay, thank you, Ms. Yuan. That is really helpful indeed. Yeah, thank you. It doesn't look like they did some challenge before the C4s. Yeah, it doesn't look like. Okay. Or actually, the challenge was the open field test. So probably the challenge was open field test.
And after this,
and after that, in 90 minutes, they did that. Okay, that makes sense. I would say that makes sense. That at least gives a context. 90 minutes. Whether 90 minutes is a good time point, that's another question, right? Because for us, we used to do half an hour, right?
By salt loading?
C4s, after salt loading or
Salt
after alpha MSH?
30 minutes.
30 minutes, yeah. So that's the peak.
Ilya, half an hour. Ilya, at half an hour time point, you have a peak in mRNA and 90 minutes protein.
Good point. That's a good point. Yeah. Actually, for the
Salt loading,
half an hour for protein, we also saw that the peak was half an hour later. At 1 hour, it was already lower, right? I think. Maybe it is dependent on the different populations. But yeah, great point. Okay. Nastya?
Okay, next question is kind of difficult. I was wondering if you happen to know the function of beta globin in the AAV vector, and also if I may ask a more general question, do you think the results of the study can be translated into improving human mental health? So regarding the first question, I think that they probably use this beta globin sequence as a control element to confirm the expression of the query.
No.
Then
they use only intron of the rabbit beta globin.
Okay,
so the sequence is the intron of beta globin from rabbit, which is known to increase in expression. So if you want to enhance the expression, there are several elements which you can introduce. It is only 400 base pairs. So we introduce that just after the promoter, before the start of the gene, and your expression will be higher because it boosts the activity of the DNA-dependent RNA polymerase. So that is a known way. It's just biotechnological way just to enhance the expression.
So then I have no clue why did they use the beta globin here.
No, I just explained.
Just because of this.
To increase the expression of
okay.
GRE and GFP. That is the key.
Okay. And regarding the second question about the mental health, honestly, I think that we are kind of very far from be able to translate these results into improving human mental health, because the first thing about human mental health is that not only some external stimuli can trigger our stress response, also some internal stimuli can do this, and we cannot check the internal stimuli like thoughts or some kind of fantasies that can also increase the anxiety level in human beings. We cannot check this on rodents because they don't have abstract thinking and self-construct.
You think?
I think so. I assume that they don't have abstract thinking yet. And the second point is that all human beings have, because we have abstract thinking and self-construct as well, we have different perceptual patterns. So the question is whether we have different perceptual patterns. Does it mean that we have different stress response? Maybe to the same stimulus. So I think these data are very...
The rats might hate you for this. For this, because they cannot tell, but they might think.
But I think they don't have abstract thinking and self-construct.
No, I don't disagree. I'm just trying to
yeah,
tease you.
yeah. Okay, so for now, I think this data is very promising, but for now we cannot apply it to human mental health. And moreover, I think that in terms of human mental health, personalized drugs should be developed, because our psyche is very different from one another, and we cannot use the same drugs to treat like some mental diseases, because as I've already mentioned, we have different perceptual patterns and different capacity to cope with stress and to like communicate with one another and so on and so forth. So yeah, I think the future is, in future maybe we will develop some personalized drugs. It will be very, very good to do this. But now we don't have them, and we cannot use this data and just apply them to cure some diseases, of course. Yeah, so any questions, comments? No. Okay. This is also kind of hard, because the quality of this figure is very poor, and I cannot understand the words, some of the words here. The data about oxytocin expression are very interesting. Please discuss this important figure. You mean this one, yeah? Yeah, it was this figure.
Yeah, I mean,
you mentioned, this S5 figure.
The first time that, or at least I don't see it often, that they publish in a good journal, they include the supplementary data in Word file, and then in a Word file with such a bad quality.
Yeah, the quality is very bad.
Like all the other data are really solid, but this is a pity.
So I don't know how to discuss
this. So I'm sorry, dear authors. Maybe the authors of this paper would hear this, see this video later. But please try to care about our eyes, and also but we don't need to really struggle to try and deciphering this very small tiny text. Yeah. If you zoom it in, it doesn't help.
Yeah, so
from here you cannot see it anyway. But if you just go and
tried to zoom it in, I couldn't manage to.
It's really bad. Yeah.
So I don't know how to discuss this. Maybe you have some suggestions.
No, I was interested in some of the targets. I was interested in something which is like mostly upregulated or mostly downregulated, but I could not, because it's really not a pleasure to look into this data. No.
Okay. Shall I continue?
Let's continue, yeah.
Okay, in figure 1, were the same rats used to analyze behavior before and after restraint stress? Yes, the same rats were used, as far as I understood. And you mentioned that in figure 1, H, PM, CRS have increased north compared to standard rats. But in fact, this tendency is not significant, possibly due to a very small number of rats. In standard group, was it just number 3?
Yes, so, this was very curious. Like they use very many numbers per group, but in this particular group, it looks like they used only 3.
6.
6?
Yeah, I wrote here. Yeah.
You're sure?
Yeah. Start from the left to right. 6, 14, 12, 6, 15, and 10 rats were used here. Yeah.
Okay.
And p-value, is
and it's still not significant.
Yeah, it's not significant.
Okay. But it is okay. There is a setting that when you show all the time all the points, that you show not in one line, but it showed dispersely. This way, it would not align on top of each other, because it doesn't look like 6. It looks like 3. But
maybe
there is a setting where you could show all of them, just also in parallel. In this regard, that would be helpful. Okay, but if they say 6, okay, we don't have any reason
I hope they don't like
to suspect that they like it. I don't think so. Yeah. Okay.
So can you please explain the grouping in figure 11? Oh, not 11. 1L. Seems complicated.
So that was complicated.
So as far as I understood, they checked the level of corticosterone before the restraint stress then on the first day of the restraint stress, then on the 7th day of the restraint stress, and then after the restraint stress. So, but I don't know why they put here RSD1, because I mean, it should be after restraint stress. It shouldn't be like...
Something's wrong.
Yeah, I guess something is wrong with the figure.
Maybe it is D7 there. Maybe the last one is D7. That would be logical, right? RSD1, then D7, then D7 again, but what is the dark gray and the light gray again? Just remind me.
Light gray is standard without restraint stress. Here, no restraint stress.
No, on the top. This before. There's also like different.
This different.
Like light gray and
I think
dark gray.
I don't know what this color.
No, that was my question. I got really confused about this.
I don't. Yeah.
So it's.
Me too. This graph looks kind of weird. And also, I didn't understand why they measure the corticosteroid level right after the restraint stress. Why they didn't like wait for maybe some days or maybe at least 1 week. They just, it seems they
mentioned.
mentioned
this data for the RSD7, right?
Yeah, but I mean, they measured it on the day 7,
yes.
And then right after, like on the day 8.
Oh.
So it's kind of weird. What's the point of measuring the corticosteroid level right after the stress, like the next day after the stress? I mean, you can wait for maybe 1 week and two, and then check if they.
That was not the point of what they studied. They wanted to see like the overall stress. I mean, the whole message is clear.
Yeah. It's clear, but it looks very.
In line with the story. Yeah. It's all so beautiful. But this grouping, to understand specifically what exactly do they mean, these 4 big groups, in each of them we have 4 other subgroups. I was really confused. It seems like they mixed up something, at least on the top. It doesn't make sense that you have twice this RSD1.
Yeah.
Somehow, maybe it's D7 there.
Yes, and also as you mentioned, we don't know when, like particularly they measure the corticosteroid level, so we cannot like.
Usually they need to write it in the methods.
But maybe I just didn't
was it the morning corticosterone or was it evening corticosterone?
Maybe I just missed it. Maybe they wrote. I'm not sure.
They should, because that is the standard way how you write about the corticosterone levels. Okay, and it was also Harry's question, so we should read very probably. Okay.
Okay, next one. So here in figure 1H, the shoreline indicating significance seems to be wrongly placed. In the text, the author claimed that PMS rats spend less time exploring novel abduction prior to RS. So probably this line should be shifted to the left. Yes, I guess you're right, but I don't know why they shifted.
It's a wrong significance.
Yeah, it's a wrong significance.
I mean, and they also write about that, but this figure is wrong. There is a wrong setting of this, wrong placement of the significance. Okay, so they need to be very careful.
Okay, here is just a comment regarding the paper's title. So yeah, I agree. I mean, it's a good suggestion. So maybe authors.
I like this title, I should say.
Yeah, this one is kind of is kind of longer, I guess. But I also like the initial title because I think
really
it's
to the
very
point. I think it really tells about the main message. I like the
and here it's just predictable maternal inspiration and adult stress resilience inside.
That is too
long. Oh, too general.
It should be more specific. I don't like this insights into this could be something some title for a book or for a thesis, but not for a paper.
Okay. Then what is the gap in previous studies? Can you please elaborate on the study background more clearly? As I see, there is a lack of linking relationship between the available diagrams. So early life stress is usually associated with an increased risk for psychiatric disorders, and while some studies suggest that early adversity can enhance stress resilience in adulthood, the biological basis of this resilience is still unknown, and maternal separation has been shown to increase the risk for depression in adult mice. However, it can also trigger some adaptive processes that improved an individual's ability to cope with stress later in life. So here they use the predictable maternal separation paradigm and unpredictable maternal separation paradigm to show the difference between these two paradigms and difference between the early life stress influence on the anxiety level and capacity to cope with stress in adulthood. So yeah, and the last part of the question I didn't really get. I'm sorry, there
It is what
lack of linking relationship.
Harry, it was
Oh,
Harry
it
was
was
asking also about
linking the
oxytocin.
available
So you
drugs.
ask
Okay, yeah. So it's like a background of the study. That's why.
It's why they went for the oxytocin. Yeah, so also.
Yeah, because it's known that oxytocin in the medial prefrontal cortex plays a key role in regulating stress, resilience, and response to early life stress. So they checked this.
That's why they went for this model and they checked that.
Okay, any questions, comments? No? Okay. Figures are overloaded with multiple factors, and also as per scientific writing, figure captions must be included below each. Picture quality can be improved because it's not good here. Yeah, I think not all of the figures are so bad, but some of them look really.
I find them quite nice, and
I mean, maybe
it was except this figure.
I think he just mentioned the supplementary figures and also this figure with wrong significance indicators. Maybe this one.
Oh, okay. But that's like not everybody picked it. So I mean, it is, and even reviewers and the editors.
But others, I like other figures
So,
as well. So
yeah,
I think they are kind of.
It's very logical. I think it's all separated blocks. I think it's very overall because.
But there are really multiple factors, but as long as you understand the paper, you can understand the figures.
Yeah,
so
it goes very well along the line of the story. So I liked it. Yeah, I agree. Yeah.
So here in figure 3, continuity is missing and PPI explanation is not included. I didn't really understand what does it mean, PPI explanation.
Continuity is missing. What is it? Continuity. It was Rizwan, right? Rizwan, you're
here
online with us. You can just comment because we don't quite understand what does it mean. What continuity do you mean?
And PPI explanation I also didn't get.
The PPI, exactly what is PPI? Yes.
Can you hear me?
Yes, we can hear. You can also switch on your video so we can also see you if you like.
Yeah.
There is one. Hello.
Yes. Can you hear me?
Yes, we can hear you.
Okay. I just wanted to know here that the continuity is missing in the picture, that we can't sum up the relationship between one, graph to the other, and here PPI mean protein-protein interaction that is basically shown in the networks. So I just wanted to know that what is the relationship between these all figures and what is the concluding in the PPI. Either it is representing the biomarker or something else.
Protein interaction.
Oh, the biomarker. Which biomarker? Sorry, I didn't get it. About which biomarker did you mean?
Yeah, I wanted to know about the network, which is highlighted red and yellow nodes.
Oh, I see.
I just wanted to know what is mean by red nodes. Either it is biomarker or what is the highlighted one mean.
Okay, this panel C. Can you explain this panel C? What is it? What does it represent?
I guess this protein interaction.
I'm not sure they checked actually protein.
Yeah, they didn't check.
Here, I think what they did is that they checked this. Basically, this heat map, they took this heat map and they checked those which are upregulated and downregulated and looked for some known predicted interactions with them, I guess. And then this is how they...
And then they identified Oxytocin, I mean, this gene.
It is not the main part, I think, of the whole thing, but the key here is to identify Oxytocin as the key. They need to have the link from the previous one, from the description to the mechanism, and they do it via this heat map.
Professor, if sorry to cut you, if they didn't, let's suppose, if they didn't want to show the interaction between proteins or half genes, or that kind of these are the nodes highlighted in red and yellow, then what is the purpose of showing this figure, this network here? I just want to know the connectivity of this figure with the heat map and the the other graph.
Are you prepared to answer that, Master? So the question is, how did they come from the heat map to the to this network?
Yeah, I'm
Yeah,
afraid
yeah.
I cannot answer.
Why they show the network? It is also represented in the heat maps. The same thing is represented in the heat map as well.
So okay, I will try to answer that. I'm not bioinformatician. Maybe some bioinformatician can help here. But as I understand, so they have found several hits, including oxytocin, and they tried to make the kind of map of interrelations of those. And they found that oxytocin is actually having, it is interlinked with very many of them. And that's how actually they needed to a legitimate reason to switch from this descriptive part towards the mechanistic part of the paper. This is how I understand. Whether they really did it this way or they already had in mind the oxytocin and they just made sequencing in order to just show it this way, I don't know. But according to their logic, this is why they did that. So they found that oxytocin is one of these central genes, including the oxytocin receptor, which is also there.
Yes.
And that's how they focused on this pathway. So this is, I think, why they have this scheme here.
Oh, okay.
At least this is my understanding.
Just to show that the oxytocin is the main,
Right.
like, let's say, main gene, kind of.
Kind of a bridge from this previous part.
But I don't know how they make this scheme. I mean, which method they use, bioinformatical or something. Yeah.
But that should be mentioned there.
Yeah, it was mentioned, I guess, but I just didn't remember. Yeah. Okay, and the second question was, figure 4, graph Q, what is shown. So yeah, it shows that overexpression of OxyTR in the mPFC had no effect on sucrose preference in UMS and sand-rat mice, rats.
Which we already discussed.
Yeah, which we already discussed before. So yeah, I think I can move on. Yeah. Okay, here, can you please briefly outline the take-home messages of figures S3, S4, and S6 from S6 to S11?
Briefly.
I think we can just briefly the graphs 3 to 4. Maternal separation has been shown to alter excitability in the medial prefrontal cortex.
No, you can't. If you do it here, but without showing the figure,
okay,
that is. You can watch here if you like. You can. Oh.
I mean, watch.
I don't have the.
Okay. I can read from my phone. Okay, and maternal stress also affects synaptic transmission in the mPFC, as evidenced by changes in spontaneous excitatory postsynaptic currents and inhibitory postsynaptic currents. And while there was no difference in excitatory postsynaptic currents frequency among the groups, the frequency of inhibitory postsynaptic currents was significantly lower in UMS rats compared to Sandra rats and PMS rats. However, the ratio of excitatory to inhibitory synaptic activity frequency was similar across all the groups. And
you're talking about which one now? You're talking about which panel?
I think I'm talking like overall, not. Here you can see this panel DEIGHJ. This one, the C from C to J. The amplitude, excitatory postsynaptic currents amplitude and inhibitory postsynaptic currents amplitude.
Can you show it with a mouse?
Yeah. This one.
Okay.
Here. I mean CE.
Yeah, but there is no difference.
Yes,
except in the D.
Yeah, so they.
Take a message is. there is a decline of the ESC amplitude. Okay, that makes sense. It's kind of going in line.
Only
something's going on with the
UMS threads.
SEPS.
Yes,
exactly.
yes,
Okay.
only this one.
Yeah. Mm-hmm.
And so yeah, I think overall these findings suggest that early life stress, like just induced by maternal separation, can lead to some alterations in neuronal.
Unless they have this kind of
synaptic
PMS, so unless they're prepared.
Unless they're prepared.
Yeah. Okay. So it all goes in line. Okay.
Okay. This one, it's for 6 and 7. So 6 is on the left and 7 is on the right. So here, I think the key takeaway from these figures is that oxytocin plays a crucial role in modulating stress, resilience, and anxiety-like behavior in rats. Yeah, so a knockdown of the oxytocin receptor in mPFC led to anxiety-like behavior in rats, particularly after restraint stress. And this finding suggests that oxytocin signaling in the mPFC is involved in regulating stress like responses and anxiety behaviors.
But also basically very similar experiments that they do.
Yeah, these figures are very similar to the main ones, so I didn't really understand why they have them in the supplementary.
Because usually there is some key difference.
Some key difference, okay.
Which I wanted to ask about. Yeah, that was my point. But okay, I mean, it looks all going really in line with the hypothesis.
So I didn't see any
okay.
differences between the main graphs here.
How many slides do we have still?
2.
2 more, okay. 2 more to go.
sorry. Okay, this one. I mean, yeah, I think you can see the take-home messages here, just briefly. The knockdown of the oxytocin receptor in the medial prefrontal cortex led to decreased excitability of pyramidal cells. We can see it. Also, they have these graphs with CIPSC and CEPSC. I mean this DEFGH. Yes. So. And oxytocin overexpression in rats exposed to maternal separation stress improves stress resilience and enhance pyramidal cell stability in the mPFC. And the effects of oxytocin.
You're not sharing the screen anymore?
No?
No.
Why?
Your sharing stopped. You're left, I think. You were kicked out.
Oh, yeah. I'm only one in the.
Hello everyone. We had some problems with Nastya, so her account was somehow kicked out of the session, but she has 2 more slides to go, and we will continue with this computer for now.
Okay. You can. How can I open the presentation?
Okay. So some technical problems. So we will resolve it. Don't worry. Yeah. So Nastya was for now summarizing the supplementary figures, and now she's trying to get back online. So she has 2 more slides to go until we finally make some kind of concluding remarks. But maybe someone would like to comment about overall story, maybe provide some criticism, some critical point, what you like, what you don't like about the hypothesis, the methods, how adequate they are, and so on and so on. Just okay. Now it should be okay.
Okay, let me. Okay. Oh no, not this one. This one.
You're muted. You're muted.
Okay. Oh, not this. How to?
Unmute.
No, unmute. But
I'll help.
Share, yeah, share my screen. Thank you.
Make also your video on.
Okay.
Okay.
Control. Okay, this one.
Sorry. Mm-hmm.
Okay. Okay, can you hear me? Is it okay? Oh, I hope. Can you hear me?
Yes.
Yeah.
Yes,
Okay.
we can
Okay.
hear.
Okay. So, yes. Oxytocin overexpression in rats exposed to maternal separation stress improved stress resilience and enhanced pyramidal cell excitability in the mPFC, and the effects of oxytocin modulation on stress, resilience, and anxiety-like behavior were specific to the mPFC and did not extend to depression-like behaviors. Yes, so I think basically
Okay.
this is the idea. And the last one. So here are two graphs. So oxytocin levels in the mPFC were found to have an inverse correlation with the corticosterone response in serum, but a positive correlation with anxiety-like behavior, as measured by duration in central zones in the open field test and open arm ratio in the elevated plasmase test. So you can see here. And yeah, so these findings align with the overall conclusions drawn from the results in rats, suggesting that the oxytocin expression is elevated in resilient postnatal maternal separation rats and reduced in susceptible PMS rats.
Wow, I should say about this graph, I think I've never seen that they list all the phenotypes that they checked and molecular findings they found in the paper, and they try to correlate with each other to see whether there is some correlation. That's kind of really brilliant. But to understand this, how does it work? Like, do you understand what is the where is the this positive correlation? I think is blue, right? And the negative is
red.
Red.
Yeah, negative is red.
So we need to check in the rows and in this diagonal, it's the same, right? So you correlate each one to each other, and then you see that mostly it is positive correlation.
I didn't understand. For example, this postnatal density is death, the last one. Yeah. So it's both. It's from
all blue except corticosterone, which is kind of opposite.
Oh,
so
okay.
Whenever you have oxytocin, kind of, or you have this, then you have this kind of increase of the amplitude and also the frequency and all this, the receptor increase. That's very interesting. Very, very interesting. Except the corticosterone. Corticosterone is kind of negatively correlating with all of them.
Mm-hmm.
It's pretty interesting way, but not easy to understand.
Yeah, of course.
Not easy to grasp all that. It's interesting. That is the last slide?
Yes, and of course, the last question, yeah. How did you find the adequacy of objectives and the way how they addressed them, the techniques used, the conclusions they came to? So I think the main, in this paper, there are two main points that can be very novel, I think. The first one is that the mPFC region is really involved into stress response, and the second one, for me, is that there is, according to this paper, a huge difference between the predictable maternal separation stress and unpredictable maternal separation stress. So if you somehow prepare for the stress in a predicted way during your early life day, it means that in adulthood you will be able to cope with stress in a more successful way. Yes, but I think maybe as I understand, they only, for example, conducted these gene knockdowns after the maternal separation and not before. So maybe it can be interesting to conduct this before the maternal separation, if it's possible, to check whether there is like influence of the level of oxytocin or not, because they only did it after the separation. I mean, so mice were already exposed to the stress, maternal separation stress, and then they conducted all the procedures, surgeries, and everything. But what if maybe they conducted it earlier? Maybe we can see something different or the same. Yes. But overall, I think this paper is very interesting and like thought-provoking, and there are so many nice results that we can like rely on and taking into account them during our own during the elaboration of our own experimental designs in future. I think it's very promising. Yeah, even though there are some like mistakes and some things that I didn't really understand maybe, but I think still it's very interesting for me.
Quite minor, right?
Yeah,
minor
yeah.
mistakes, I would say. Yeah. Okay, great. Maybe I can start sharing.
Yeah,
screen,
of course.
If you don't mind.
Yeah.
Okay, I have several points here to mention, which I find very interesting. If you allow me, let me share my screen. Share my screen so that let me try sharing my screen so that you would. Okay. Wait. Share my screen. I will share this window. The whole entire screen. That one. Okay, and then share. Oops. Okay. Good. Then switch here. Okay. Look at this. I would like to draw your attention to this interesting finding. They found that oxytocin levels are changing if they manipulate the receptors. Was it here or was it in the previous? What was it? Wait a minute. Just let me see. Is it this figure where they manipulated the receptors? I don't quite remember. Where they find that oxytocin levels are also changing, and they discuss it just briefly. Let me see. Lassie, can you remind me, please? Which figure was it when they manipulated the receptors?
Okay, let me figure number.
Oxytocin.
Number 4, I guess.
4, maybe 4. Yeah. Maybe I'm checking the wrong one. Yes, yes, and here they also checked oxytocin. Yeah, look at this. So they, in those which are having the oxytocin receptor expression, increased oxytocin receptor, you see? They see also increased oxytocin, and vice versa. If they use, if they block oxytocin receptor, they see lower oxytocin. It is not very straightforward to understand why would that happen. They explain that they suppose that there is like feedback regulation. So if you have too much of the oxytocin receptor, then it would there would be a feedback to also increase. If you block it, then you would have like decreased blocking of the presynaptic oxytocin receptor. So kind of oxytocin would be mostly so if you increase the oxytocin receptor in the postsynaptic neurons, then it would be bound to this postsynaptic, but not to the presynaptic.
Okay.
And then it would like signal to feedback much less than it would normally. And that would per se also increase the expression of the oxytocin. That's what they show in this figure 4O, as well as opposite phenomenon on the figure 4D. I think it's very interesting to show.
Yeah.
And it is not actually very obvious, not very straightforward. So this is one point which I wanted to draw attention to. And the other point is about this, the paradigm which they used here in the figure, it is 5, right? Yes, figure 5.
Yeah.
Do you understand this paradigm? Look at this. I think it's very interesting. And it is, I think, the key experiment. Do you understand how does it work? Because they injected this Cre virus to this to the prefrontal cortex, so that it is retrogradely transported back to the PVN.
Yes.
And then this Cre would induce this recombination in this DREAD, in this HM4D. So by that they only target those neurons which are oxytocin, because this DREAD is expressed on the oxytocin receptor, oxytocin promoter, but also dependently on the projection to this
mPFC.
yes, prefrontal cortex. So only those oxytocin neurons that are projected to the prefrontal cortex would be inhibited here. So that is, I think, very elegant way to show like this. This is, I think, the key experiment.
The technique is very.
Yeah, that's
But did they show that? Did they show that? Because on the scheme you do not see this Cre virus. It was also a bit strange for me. They show only GFP virus.
Exactly. So this is exactly my point. When I first heard the presentation of NASA, I also didn't understand that. I needed really to go deeply into this paper, and they explained that actually they express CMV, beta globin, Cre, which is missing here. So that's also missing in the scheme. There is a Cre P2A GFP, and the Cre is most important here. So Cre would be transported back, and they explicitly say that it's retrogradely targeting AAV, so it will be uptaken to these neurons which are downstream, and then they will be reuptaken back to the PVN, and only those neurons which actually project to the PVN would in the end be inhibited, which is really cool. But they didn't nicely show it, not in this scheme, and also they mentioned it just briefly in the paper. So so I would also like to draw attention to this one, because that's a key experiment. It's really great, really nicely designed one, but they didn't describe it nicely. Even in this scheme, what Vitek says, Cre is missing. They don't write it as a Cre, but it is actually a Cre virus. They showed in the paper. Yeah. Okay. So from my side, I agree with Nastia. It's really solid data. I don't actually find anything disturbing here. Really logically done. Like you show all the loss of function, gain of function. You have like really nice storyline. Really well designed. Really nice paper. Yeah, I like it very much. Any other opinions? Any other comments, please? Anyone? Okay, I hear no comments so far. So then, if there are no comments, then I would like to thank everyone again. I think it was really great discussing with you. Really nice, really nice feedback from all of you, and I think also it worked really well. I really liked the discussion. Yeah. So thank you very much, and we're looking forward for our Polish colleagues to present the next paper. So you please prepare a presentation, and you can send the video. We will send a link where you can send how you can send the video to us. And if you have any questions, just ask me.